Thank you for that introduction, John. I'm very happy to talk to you all today about what I'm going to be doing with the minimal cell. So, a lot of scientists have been interested in this question: what is life and how did it originate? From an evolutionary standpoint, it is thought that life arose from a universal ancestor that, after years of evolution, gave rise to organisms and these three major domains of life. Earlier in evolution, the first set of cells that arose on Earth were thought to live in what they call the primordial soup, which is essentially an environment where there were very simple molecules that these cells that were there during this time were somehow able to take into their cells and undergo simple types of metabolisms and undergo basic replication to be able to grow and survive. But we don't know exactly what these cells look like and how they were able to do all of this stuff. So JCVI set to do this by creating a minimal cell, as many people in this audience are familiar with. Not so much from an evolutionary standpoint, but from a genomic standpoint. JCVI gets its name from anything that's genomics related. So the creation of a minimal cell started with SIN 1.0, which has 901 genes. JCVI was able to get that down to SIN 3.0, which only has 473 genes. And when SIN 3.0 was first constructed, there were 149 genes of unknown function. It seems rather reasonable to assume that if you were to streamline a bacterial genome to its minimal components, at least I would think that we would know what the function of most, if not all of what those genes are. But it turns out that we don't. So there's really a lot about cell biology that we really don't know at a very basic level. So 149 genes, we were able to get that down to 80. So we still have 80 genes of unknown functions. And a lot of what I do on a minimal cell is based on me characterizing a lot of these genes with unknown functions. And I'm going to talk about a few of those genes today. So one of these genes that James talked about a few moments ago in his talk, gene 33, which he hypothesizes and calls for a probable lipid transporter that may have a role in cell division. This is um, the structure of P116, the product of MPN213 and M. pneumoniae, which is thought to have this hydrophobic cavity that binds to a lot of different types of lipids and excludes others. It also has these two N-terminal domains that are thought to face outside of the cell and can sort of switch orientation when they bind specific lipids. So one of the things that I'm working on to address this question as to whether gene 33 is a lipid transporter is that we're going to put this, what we're going to do is replace gene 33 with the M. pneumoniae gene. So one of the things that's worth mentioning is that similar to M. pneumoniae, in which gene 213 is essential, gene 33 is also essential in our minimal cell. So the idea here is that if we were to delete gene 33, where we should not recover viable bacteria, and replace that with this gene from M. pneumoniae, and we're able to get viable cells, it's reasonable to assume that the function of this M. pneumoniae gene is able to substitute for the loss of function of gene 33, and that suggests that it might be a lipid transporter. So I've created a piece of DNA that has this M. pneumoniae gene that's continuous with the minimal cell genome. And one of the things that I'm doing is putting this into yeast. So we have a Saccharomyces cerevisiae strain that constitutively expresses Cas9 and has our minimal cell genome on a centromeric plasmid. And what I'm doing is deleting gene 33 and then providing the M. pneumoniae gene. And then in a transplantation experiment, put that into M. capricolum. We heard Phelan last Friday talk about genome transplantation a lot. A lot of the other genes that are uncharacterized in the minimal cell genome, James also talked about this as well, are genes that are associated with cell division. So SIN 1.0 forms a lot of spherical shaped cells, whereas SIN 3.0 forms a lot of irregularly shaped cells. One of the things that I'm working on in collaboration with the Luthi Schulten group at Illinois and the HaLab at Harvard University is looking at the cellular localization of FTSZ. As mentioned before, FTSZ is a protein that forms a polymer at the division site of a bacterial cell. So I created a piece of DNA where I put M-maple, which is a fluorescent tag on FTSZ. And we're also interested in looking at DNA-A. So before cells fully undergo cell division, cells have to replicate their DNA. DNA-A is a protein that binds to the chromosomal origin of replication, and that then recruits other replication proteins that facilitates DNA replication. So I've also created a piece of DNA that has M-cherry in the middle of DNA-A, and I'm putting this into our minimal cell. And with our collaborators, we're going to look at these cells using high resolution fluorescence microscopy. So we are doing this using Cre-Lox recombination. So we have a version of our minimal cell that we call Syn3B, which is essentially Syn3A that has LOX-P sites in its chromosome. And what I'm doing is putting these constructs on a plasmid that has Cre recombinase. Once I introduce that plasmid into the minimal cell, Cre recombinase is going to facilitate DNA exchange between these sites, and that's how we're going to get these genes into our minimal cell. Another thing that I'm looking at is the chromatin structure of our minimal cell. So in the evolution of cells, particularly in cells with very large genomes, in the evolution of eukaryotic organisms, there was a relatively large genome expansion. In order to be able to pack all of this large genomic DNA into a cell, cells evolve ways to do that. If we're talking about eukaryotic organisms, we're talking about the nucleus. So there's chromosome proteins that bind to DNA to facilitate the packing of all of this. And this represented a major step in the evolution of organisms. So by looking at chromosomal structure, we might be able to learn a lot about the origins of life. So Ben talked about this earlier today. There's this method called 3C mapping, which is a method that people use for looking at chromosome structure. Essentially, you cross-link cells with something like formaldehyde. You separate those cross-link fragments, you ligate them, and then you can sequence them to look at points of contact. So when we're talking about chromosomal contact, we're talking about nucleotides that physically are 1,000, maybe 1,000,000 base pairs away, but are actually physically interacting with each other to be able to pack all of this DNA into a cell. So the output of this data is a contact map that has the genomic coordinates on the X and the Y axes. And this diagonal line represents the chromosome. There's a heat map that represents which regions on the chromosome are interacting. And the greater the intensity on the contact map, the greater the extent of those interactions at any given site. So this is a contact map that's been generated in E. coli, which exhibits a lot of chromosome interactions. When this experiment was done in collaboration with Remus Dame in the Netherlands, to our surprise, we did not see any chromosome contacts in our Syn3A minimal cell, which was very surprising. If you take a closer look at the genomes of Syn1 and Syn3A, both Syn1 and Syn3A have this gene called HUP. HUP is a gene that encodes for a bacterial histone-like protein. HUP-A, which is present in many different bacterial species, can bend DNA and can relieve negative supercoiling. The effect of this HUP-A interaction is that you can significantly influence which genes are expressed at any given time in a cell. One of the things that's interesting to us is that upstream of HUP-A, Syn1 has a gene called GPSA that appears to be absent, that's definitely absent in Syn3A. So we think that we accidentally messed up the expression of this histone-like protein and the genome minimization process. So we're interested in better exploring the role of HUP-A in maintaining the chromosome structure in our minimal cell. So to do that, what I'm doing is adding GPSA back into our minimal cell, and then we're going to do a 3C mapping experiment with that in parallel with Syn1 DNA. And then we're also interested as to whether HUP-A is essential in our minimal cell. So you can delete HUP-A and E. coli and recover viable bacteria. They look very weird. They grow relatively slow, but you cannot delete HUP-A and B. subtilis, for example. So we don't know if HUP-A is essential in our minimal cell. And if it's not essential, what is the chromatin structure and that cell looks like? So we essentially think that we have recapitulated a cell that looks like one that emerged very early in evolution, specifically before cells develop complex ways to divide their DNA and develop a way to organize chromatin structure. And we hope that a lot of these experiments will shed some light on that hypothesis. And then with that, I would like to acknowledge a lot of labs at JCVI, Yo's lab, aside from the glass lab, and all of our collaborators. And I'm open to taking any questions. Thank you for listening.
Questions? Ronald, how long before you think we'll be able to get some answers on these things?
Which part? All of the proposed experiments or just a few of them?
You pick one. What's the first thing we're going to know about?
Let's see. So the FTSZ DNA A labeling, I would say we can send cells out hopefully within the next 2 weeks, week and a half, two weeks.
Mm-hmm.
And the HUP-A stuff, I would think a little bit longer because transplantation is a little finicky still. So Phelan, me and you're definitely gonna need to have a more in-depth conversation about that. But yeah.
And so you and I have talked about this, replacing the possible lipid transporter in SYN3A with what has been suggested to be a lipid transporter in pneumonia. How close do you think, is that good enough to say that that's what this gene is doing, or is that still, do we need more?
I think it'd be nice to have more. I think if we can replace the gene and get viable bacteria, it's reasonable to assume that they serve the same function, but I think there's definitely additional experiments that we can do that will definitively state that it is a lipid transporter, maybe doing like a lipidomics experiment of some kind. I think replacing the gene is good enough evidence, but not all the evidence that we need to say it's a lipid transporter, but very likely the case if we get the result that we expect.